Namaste friends, welcome back to Biology Center. Today I came up with a new concept from the genetics, that is gene interactions. Today's topic for our discussion is gene interactions. What are gene interactions? The expression of a single character by the interactions of more than two pairs of genes is called gene interactions. Here the single character is going to be produced because of more than two pairs of genes interacted. So in case of some Mendel experiments, so what we saw is so one trait, so one gene concept. So one trait, one gene. One gene is going to influence the one character. But in some other animals, in some other organisms, so that case was not true, because the Mendel did not did the same experiments in all the organisms. There are some deviations. There are some deviations from what Mendel said. So exactly, so there are deviations from the Mendel inheritance pattern. So here the gene interactions are the extensions to the Mendelian genetics. These are the extensions to the Mendelian genetics. So simply I can tell you, so more than two pairs of genes are going to influence a single character by their interaction. Right. So here, so one hypothesis, name of the hypothesis is called factor hypothesis. Factor hypothesis. It was given by Bateson and Punnett. The factor hypothesis was given by Bateson and Punnett. So what this hypothesis says? Some characters produced by the interaction of two or more than two pair of genes. This is the factor hypothesis. In some organisms, some characters are produced not by just interaction between a pair of genes or single gene. These characters are produced by interactions between more than two or more than two pairs of genes or factors. So let me tell you types of gene interaction. So already I mentioned one point that gene interactions are the extensions to the Mendelian genetics. So because of many deviations were observed by other scientists. So let us take one chart, brief chart. These gene interactions are two types. The first type is allelic gene interactions. Allelic gene interactions, they are also called intra-allelic gene interactions. The second type of gene interactions is non-allelic gene interactions. These are also called interallelic, right? Interallelic gene interactions. So before going to take examples for both types, so let us know, so what is the meaning of allelic gene interaction and non-allelic gene interaction. Here, the allelic gene interaction means interaction between two alleles of a same gene present in the same locus on same chromosome. So, right, we have the homologous pair of chromosomes. Here, the allelic interaction takes place. Two genes, right? Two genes present in the same locus of same chromosomes. Here, the interaction between these two. So this allele interact with this allele interact with this, and express the particular character. So we saw in the monohybrid cross of Mendelian experiment. Next, come to non-allelic gene interaction. Here, the interaction takes place between two genes present in different locus on same chromosome or different chromosome, right? Here, two genes are involved in non-allelic gene interaction. They present in different location, not on the same locus, right? So they present or they are located in the same locus on the same chromosome or different chromosome. So let me list out examples for allelic gene interaction and non-allelic gene interaction. So examples. The first one, complete dominance. We saw this case in monohybrid cross and dihybrid cross of Mendel experiments. Second one is incomplete dominance or blending of inheritance. Another name for incomplete dominance. Complete dominance, incomplete dominance, then multiple alleles or multiple allelism. The fourth one is pleiotropy. So one more can also add here, that is lethal genes. These five are the examples for allelic gene interaction. So next, so we take examples for non-allelic also. The first one is complementary genes. Second one is supplementary genes. Third one is duplicating genes. The fourth one is epistasis. So it may be dominant or it may be recessive. Dominant epistasis and recessive epistasis. So it is about the meaning of gene interaction and types of gene interaction. So let us discuss each type of gene interaction in detail. The first we talk about incomplete dominance. Incomplete dominance. So before going to that, we need to remember the Mendelian inheritance pattern. So how was the Mendelian inheritance pattern? There are two alleles of a gene, or there are two copies of gene as a pair of alleles. So one is dominant, another is recessive. So always dominant allele completely dominant over the recessive allele. That was the case. So we saw and understood from the Mendelian inheritance pattern. Now, so we move towards the deviations. So from the Mendelian inheritance pattern. So let us see how. The first genic interaction is incomplete dominance. Incomplete dominance. What is incomplete dominance? Simply dominant allele incompletely dominant over other allele or another allele. It is called incomplete dominance. This concept was actually explained firstly by the person Correns. So name of the scientist who was explained the incomplete dominance type of gene interaction was Correns. He did experiments. Experiments in the plant Mirabilis jalapa. So which is also called 4 o'clock plant. 4 o'clock plant. So here itself I give other examples also. The one example is Mirabilis jalapa. The another example is Antirrhinum majus. It is called snapdragon. Antirrhinum. So majus. Another one is snapdragon or dog flavor, right? So these are the three examples for it. So let us understand the incomplete dominance by taking the example Mirabilis jalapa. For the experiments, parent generation was taken. So one 4 o'clock plant that is having red color flower, that is RR. Another plant is white flower producing plant. Okay na? This produces red color flowers. This produces white color flowered plant. Next, write the gametes. This plant produces all the gametes containing capital R allele. This plant produces all the gametes containing small r allele, that is recessive allele. Now we do the cross pollination, right? So we do the cross pollination, right? So all the offsprings we got in the F1 generation are heterozygous plant. This is the F1 generation, but the character expressed by this hybrid was pink color. This is the pink color flower giving plant. Pink color flower giving plant. Here the color pink is the intermediate color between red and white. So by this, what we can understand here is the R, the dominant allele R, incompletely dominant over the recessive allele, that is small r. Because of that reason, because of incomplete dominance by r over the small r, the pink color, that is intermediate color, so we got. Next, continue the experiment further. Next, do the selfing between F1 generation. Take RR into one more RR plant. This is the pink flower giving plant. This also pink color flowering plant. Then write the gametes here. It produces two types of gametes, with some are with capital R, some of the gametes with small r. This plant also same, capital R, some gametes have capital R, some gametes have small r. Next, do the crossing. This gamete may have chance to fuse with this gamete. This gamete also have chance to fuse with this gamete. Then this gamete have chance to fuse with this gamete. Then this gamete also have chance to fuse with this gamete. Now write the genotype. So of course, instead of this way, so we can draw the Punnett square also. So we can follow either of them. This one, capital R, then capital R, small r, then capital R, small r, then small r, small r. So write the phenotypes. Capital R, parent type, red color flowering plant. This capital R, small r, so hybrid one, that is a pink color producing plant. Again capital R, small r, pink color flowering plant. Next, smaller parent type, white flowering plants, white color flowering plant. So here, so what is the phenotype ratio and what is the genotype ratio in this experiment? First write phenotype ratio, then genotype ratio. So what is the phenotype ratio? External appearance, right? Red color is 1, 2 pink colors, 1 white color. Then we can write it as 1:2, 2:1. Then genotype ratio, this is the one type, RR. These two are having same genetical constitution, that is capital R, small r. The last fourth plant has a RR as genetic constitution. Again the ratio was 1:2, 2:1. So here, so we need to make a keen observation. So what is the phenotype ratio and genotype ratio of Mendel? The phenotype ratio of Mendel is 1:3. The genotype ratio is 1:2:1. Here the phenotype ratio in this experiment deviated from the Mendelian phenotype ratio. Mendelian phenotype ratio. So by this result, so we can say that in some organisms the dominant allele incompletely dominant over the recessive allele. Because of that reason, so we got different organism, I mean intermediate character producing hybrid organism in the F1 generation. So here so one more example I would like to give you from the animal side. So if we do the same experiment in snapdragon flowering plant, so we receive the same ratios, 1:2:1, 1:2:1 ratio. So another example for incomplete dominance from animal side. Feather color in fowl. First, so we take the parent generation, black feather fowl, that is capital B versus white color, that is small b. So it is black color feathers. So this fowl has white color feathers. Then write the gametes here, capital B. So here small b. Then cross fertilization, cross pollination. All F1 we got here heterozygous having capital B and small b. This hybrid is going to be blue color feathered fowl. So what are the characters exhibited by both the parents? This parent exhibits the black color feathers. This parent has white color feathers. So when we cross pollinated these two parents in the F1 generation, the hybrid we got is intermediary one. This is the intermediary one because it expresses the character blue. Here the color blue is the intermediate between black and white color. Then continue the experiment further. So do the selfing between F1 hybrids BB into BB. So here I would like to have the Punnett square. Instead of draw the lines, this is the Punnett square. So take the female gametes vertically, then take the male gametes horizontally, then write the combinations, capital B, small b, then capital B, small b, then small b. So this is the black feathered fowl. So this is the blue feathered fowl. This also blue feathered fowl. This is the white feathered fowl. Then so what is the phenotype ratio? So what is the phenotype ratio? Then what is the genotype ratio? Phenotype ratio externally how they are looking. So one block, 2 blue, 1 white. Then 1:2:1 is the phenotype ratio. So what is the genotype? So one has capital BB as the genetic constitution 1. So 2 fowls have capital B small b genes, that is again number is 2. Then only one has 2 recessive alleles, then 1. Here also in this experiment also both phenotype ratio and genotype ratio are same, that is 1:2:1. This phenotype ratio is deviated from the Mendelian phenotype ratio in the monohybrid class, that is 3:1. So it is about the incomplete dominance. Thank you for watching this video.